Good morning, students. Myself, Harpreet Boparai, lecturer biology, posted at Government Senior Secondary School, Pumbli, District Gurdaspur. Today, I am going to discuss immunity and its types. Contents of today's topic are immunity, types of immunity, different types of barriers and cells for immunity. Difference between innate and acquired immunity. Difference between active and passive immunity. Structure and different types of antibodies. Antigen antibody interactions. Humoral and cell mediated immune system. Now, first of all, I am going to tell you about the basic concept of immunity. What is immunity actually? Immunity is the ability of body to protect the body from all type of pathogens, means disease causing organisms like bacteria, virus, toxic substances, etc., which enters the body. Immunity is also known as disease resistance. Lack of immunity is called susceptibility. Now, different types of immunity. Immunity is of two main types. First one is innate immunity and second one is acquired immunity. First of all, I will discuss innate immunity. Innate immunity is also known as inborn or natural or non-specific immunity. As the name inborn indicates, this immunity is inherited from parents, and non-specific name indicates that this immunity is not specific. It provides immunity from wide range of pathogens. Innate immunity provides protection throughout the life. Example: humans have innate immunity against distemper, which is a fatal disease of dogs. Now, I am going to discuss different barriers which provide innate immunity. First one is physical barrier, which includes skin and mucous membrane. Skin's tough layer, stratum corneum, prevents the entry of bacteria and viruses. Mucous membrane provides mucus, which traps the microorganisms and dust during respiration. Now, the second barrier is physiological barriers, which includes skin, mucous membrane, body temperature, pH of body fluids, etc. Sebum inhibits the growth of microorganisms on skin. Lysozymes present in tears and saliva dissolves the cell walls of microorganisms. Mild fever strengthens the defense mechanism by activating phagocytes and inhibiting growth of microbes. Now the third barrier is cellular barrier, which includes neutrophils, monocytes, macrophages, and natural killer cells. Now the fourth barrier is complement system. Complement system consists of 20 different type of proteins which provides protection from infection. Complement proteins creates pores in the plasma membrane of microbes. Complement system destroy the microbes by cytolysis. inflammation, and phagocytosis. Now, the fifth barrier is inflammation. It is a defensive response of body to tissue damage. The signs and symptoms include redness, pain, heat, and swelling. Now, the last is cytokine barrier, which include interferons. Interferons protect the body against viral infection. Now, come to the acquired immunity. Acquired immunity is also known as adaptive immunity or specific immunity. This immunity is acquired by an organism after birth. So it is called acquired immunity. Acquired immunity is able to identify different types of foreign molecules. Acquired immunity is able to recognize self and non-self molecules. In acquired immunity, immune system has memory of first encounter of pathogens. Acquired immunity is of two types. First one is active immunity and second one is passive immunity. Now the first one, active immunity. When organism's own cell produces antibodies, when they come in contact with antigen or vaccine. Active immunity takes time for formation of antibodies. Active immunity is long lasting and harmless. Example: person suffering from smallpox develops active immunity. Now the second type is passive immunity. Immunity is said to be passive when antibodies produced in other organisms are injected into a person who already has a potential antigen in his body. Passive immunity provides immediate relief and this is not long lasting. Active immunity has two types. First one is natural active immunity and second one is artificial active immunity. Now, natural active immunity. During natural active immunity, antibodies made after exposure to an infection. Example: person suffering from smallpox develops natural active immunity. Now, artificial active immunity. During artificial active immunity, antibodies made after getting a vaccination. Example: polio vaccination leads to the formation of antibodies. Now come to the passive immunity. Passive immunity has two types. First one is natural passive immunity and second one is artificial passive immunity. Now, natural passive immunity. Immunity transferred from mother to the fetus through placenta. Up to 3 months of age, a child is protected by mother's first milk, having IgA antibodies. Now, artificial passive immunity. Immunity is provided when antibodies produced in one organism are inserted into the organism infected with the same antigen. Example: antibodies against snake venom. Now, difference between innate and acquired immunity. Innate immunity is also known as inborn immunity, and acquired immunity is acquired by organism during life. Innate immunity generates non-specific immune response. Acquired immunity generates specific immune response. Innate immunity forms the first line of defense of immune system, while acquired immunity is the second line of defense. It is a action against pathogens that overcome innate immune defense. Innate immunity generate a rapid response. Acquired immunity takes 5 to 6 days. Innate immunity has plasma protein, phagocyte, physical and chemical barriers, while acquired immunity have two components, humoral and cell-mediated immunity. Now, in case of innate immunity, this immunity does not generate memory cells, while acquired immunity generate memory cells. Now, difference between active and passive immunity. Active immunity is produced due to contact with pathogen or antigen. Passive immunity is produced due to antibodies obtained directly. Active immunity is long lasting, while passive immunity lasts for few days. Active immunity has no side effects, while passive immunity may cause side effects. Active immunity is slow in action and provides slow relief, while passive immunity is fast in action and provides immediate relief. Active immunity generates memory cells. Passive immunity does not generate memory cells. Active immunity produces antibodies by the infected organisms. In case of passive immunity, antibodies are not produced by infected organisms. Structure of antibodies. Antibodies are also known as immunoglobulins. Immunoglobulin is a large and complex molecule. It is composed of 4 polypeptide chains, 2 large identical heavy chains, and 2 short identical light chains. These 4 chains joined into a Y shape by disulfide bonds. Each polypeptide is composed of constant and variable sequences. A light chain consists of one constant sequence and one variable sequence. Heavy chains consist of one variable sequence and three constant sequences. The variable sequences are located at the distal ends of Y arms. Different types of amino acid sequence on variable region provides specificity to the antibodies. There are 5 types of antibodies. First one is IgG. This is most abundant class of antibody, forming about 80% of total antibodies. IgG is able to cross placenta from mother to fetus. Now the second major type of immunoglobulin is IgA. It is second most abundant antibody, forming about 10 to 15% of total antibodies. It is found in sweat, tears, saliva, mucus, and colostrum. Now, the third antibody is IgD. It forms only 0.2% of total antibodies. These are present in intestinal fluids of normal persons. Now, the fourth type of antibody is IgE. It forms less than 0.1% of total antibodies. These are present on mast cells and basophils. It may prove more harmful than useful to a person who is allergic to a substance. Now, the fifth type is IgM. This forms about 5 to 10% of total antibodies. It is the largest antibody secreted by plasma cells. Fetus is capable to synthesize this antibody. Antigen antibody interactions. The antibodies fight the antigens in 5 different ways. The first way of fighting is neutralization. Some antibodies called antitoxin combine with antigen termed toxin and neutralize their action by masking their harmful groups. The phagocytes then dispose of the neutralized antibody antigen complexes. Neutralization reaction eliminates the harmful effect of virus or exotoxins. Now, the second interaction is agglutination. Some antibodies cross-link antigen into groups for easy disposal by phagocytes through ingestion. Now, the third interaction is precipitation. Some antibodies cross-link the molecules of soluble antigens to form an immobile precipitate which is easily ingested by the phagocytes. Now, the fourth interaction is opsonization. Some antibodies coat the surface of microbes so that these microbes are easily killed by phagocytosis. Now, the fifth interaction is complement activation. Antigen antibody complexes activate complement components of acquired immunity. First one is humoral immunity or antibody mediated immune system, that is also called AMIS. And the second component of acquired immunity is cellular immunity or cell mediated immune system, that is also known as CMIS. Humoral immune response is mediated by antibodies. During these responses, immune system triggers specific B cells to proliferate and secrete large amount of antibodies to destroy the pathogens. Immune response begins as the antigen enter the body. The immune system trigger the specific B cells to produce antibodies. The humoral immune response begins with activation phase. During activation phase, a macrophage, one of the cell of immune system, engulf the invader antigen through phagocytosis. Inside the macrophage,
the new vesicle formed, called phagosome, which fuses with lysosome. Then digestive enzymes breaks the antigen particle. These antigen combine with class 2 MHC molecules and move to the outer surface of macrophage. Macrophage with antigen fragment on outer surface is referred to as antigen presenting cells. Another cell, T helper cell, recognize the antigen on the antigen presenting cell and then become activated. The receptor on T helper cell bind with MHC class 2 receptor and antigen particle inside it. After that, macrophage release chemical cytokine interleukin 1 which stimulate T helper cell to secrete interleukin 2. The activated T cells divides and increase in number. These T cells stimulate B cells which will differentiate into plasma cells. The plasma cells produce antibodies in large number. These antibodies bind with antigens and destroy them. The second major component of acquired immunity is cell mediated immune system, that is also known as CMIS or T cell immunity. This system defends the body against viruses, fungi, and some bacteria. The cells provide immunity in this system are T lymphocytes. Cell mediated immune system is explained by an animation. Major cells of cellular immunity are T lymphocytes. T lymphocytes develop in thymus. During maturation, variations arises in T cells due to different types of surface proteins. These surface proteins are called T cell receptor. Helper T cells has receptor CD4 and second population of T cell is cytotoxic T cells having receptor CD8 during maturation. T cells learn how to recognize self and non self cells. T cells migrate to lymph nodes and other lymphoid tissue where they come in contact with different types of pathogens. T cells can only recognize those antigens which are displayed by antigen presenting cells on MHC class 2 receptors. There are two classes of MHC molecules. MHC class 1 molecules are expressed by all nucleated cells of body. These molecules are continuously produced in cytoplasm. They pick the pieces of peptides and displayed them on surface. If cell is affected with virus or cell is cancerous, then the antigens are displayed by MHC molecule on surface of antigen presenting cell. MHC class 2 molecules occurs only on professional antigen presenting cells like dendritic cell. Dendritic cells pick up antigen and cut it into pieces and display them on MHC2
molecule on the surface. Then these dendritic cells migrate to nearby lymph nodes where they present the antigen to matching T helper cells whose CD4 receptors bind to MHC2 molecules. Activation of T helper cell require the second binding between T cells. Activated T helper cells divides by mitosis which leads to clonal expansion. Cells produced through mitosis are specific for pathogen. Some of the cells differentiate into effector cells and other become memory cells. Most of effector cells leave the lymph nodes through blood vessels and migrate to the site of infection where they attack the pathogen. Helper T cells produce interleukin which lead to activation of T killer cell, B cells and other cells of immune system which have diverse functions. Killer T cells are the main cells of cellular immunity. They release toxins and directly kill the affected and cancerous cells. Effector cells dies after infection is cured, but memory cells live much longer time. They reside in lymph nodes and circulate in blood and migrate to peripheral tissues. Memory T cells are more numerous than naive T cells. If body is exposed to same pathogen, the memory cells provide much faster immune response, so no sign of illness is seen. Suppressor T cells have regulatory function. When infection is cured, they inhibit proliferation of effector T cells. They also differentiate between self and non-self cells. So in this way, they protect from autoimmune diseases. So this is all about immunity and its types. I hope you understand the concept. Thanks.